Anyways, I'm going to talk today about our studies on the role of the endocannabinoid system in the transition from acute to chronic pain. So this is a little bit of follow-up, or is related to my previous work that I showed yesterday. So these are the learning objectives that you probably don't care, and it's okay, because I don't care either. And this is another slide we can skip, because this is just explaining what pain is. It's very difficult to define pain, because it's very subjective, right? It's nothing that we can actually describe. If I ask you, or if an extraterrestrial come and ask you, so what is pain, it's going to be very difficult for us to explain what pain is. But the International Association for the Study of Pain defines it as an unpleasant sensory and emotional experience associated with actual or potential tissue damage, or described in terms of such a damage. That means that we don't understand what is pain, right? Because I don't know what they want to say with that. But anyways, painful is horrible, you don't like it, and normally it's because this is a protective phenomenon, right? It protects us from damage and protects us for potential tissue injury and further damage. But pain in some occasions is not necessary anymore. For example, in chronic pain conditions, when we know why we have pain and we need to fix that situation, we don't need pain anymore. People don't need to be in pain if we know the cause and if we want to treat it. So chronic pain definitely deserves treatment. And as I said yesterday, we don't have effective treatments to manage chronic pain. We are doing very, very badly on that. The spinal cord is a very important organ and area, anatomic area for pain management, for pain mechanisms. The reason is because pretty much everything that happens in terms of neuronal activity or physical activity and also molecular activity for pain conditions has to pass through the spinal cord. That means that if we actually have pain in our extremities or in any of our foot, hands, or whatever, will travel from nociceptors to the spinal cord. In the spinal cord, this information will be, let me see if I can manage this. The information will travel from the periphery to the spinal cord, and here in the spinal cord, this information will be processed by interneurons and by communications from other neurons, and also glial cells, microglia, astrocytes, prevascular cells, etc. This information will be modulated at that site. And then from there, it will go to the periphery and do the withdrawal reflex, the contraction of the muscles, and then also this information will go to the brain. And in the brain, when that information reaches the brain, at that moment, we can actually say that this is pain, because we are conscious that this is happening. Memories and also stress, other conditions, etc., emotions will also modulate pain information. So then this information will go down as well from this brain to, again, the spinal cord, and this information will be also remodulated again. And then it will go to the periphery and it will go up. So in the spinal cord is a very important organ, and pretty much the drugs that we have right now, the better drugs that we have for the treatment of pain, they work at the spinal cord level and at the brain, so in the central nervous system. Cells that work only at the periphery have limited effect for particularly central pain, but that's why we want to have drugs that penetrate the blood-brain barrier. However, as you can imagine, the side effects for these drugs could be also an important issue. These are some numbers. I gave you yesterday some numbers. Chronic pain affects more than 116 million people in the United States, and it costs over $600 billion, more than cancer and diabetes together. Surgery also is one of the large sources of chronic pain. Up to 50% of patients may develop this. About 48 million surgeries are taking place in the United States, and again, the mechanisms for acute to chronic pain are not understood. So I'm going to try to connect the endocannabinoid system with the transition from acute to chronic pain, or in the mechanisms that underlie the resolution of acute pain after surgery. So just as a reminder, what is the endocannabinoid system? We have two major endocannabinoids. That means ligands to the receptors of cannabis, or cannabinoids, or THC. These are anandamide and 2-AG. Sorry for the small font here. But it's anandamide and 2-AG are the two major endocannabinoids. They act similarly to THC, for example. These endogenous cannabinoids work by binding and activating cannabinoid receptors, CB1 and CB2 receptors. CB1 receptors, or cannabinoid type 1 receptors, are mostly expressed in neurons in the central nervous system. And CB2 receptors are mostly expressed in the immune cells at the periphery. But also, our laboratory has shown that they are expressed in the central nervous system, mostly in microglial cells, so preserving their immunomodulatory function. And then we have enzymes that degrade endocannabinoids. These enzymes degrade anandamide and arachidonyl glycerol, or 2-AG, and therefore the endocannabinoid signal is shut off by these enzymes. As you can imagine, these are all of them targets for drugs. And in fact, we have inhibitors of these enzymes that will enhance the endocannabinoid tone and therefore provide some potential therapeutic effects. All right, so what about endocannabinoids and pain? We have already a couple of talks about the endocannabinoid system and pain modulation. So in general terms, the endocannabinoid system or the cannabinoid receptors are actually circuit breakers for pain signaling in general. Endocannabinoids produce in general, with some potential details, but in general they produce a reduction of the activity of nociceptors that is translated in a reduction of pain-related behavior in animals, and in humans, like marijuana, for example, has some effects in some types of neuropathic pain. It has been described that this endocannabinoid system is produced in demand, as we heard yesterday as well. That means that if we have an inflammation going on in a particular part of our body, the endocannabinoid system will react only at that part, not in the whole system. So that's a very important point. Well, anyways, I have more information here, but I think it's not that relevant for my lecture. So again, this is just a review of the type of cells that we have in the central nervous system, particularly in the spinal cord, cells that have immune capacities, obviously microglia, which are the macrophages of the central nervous system. We have also astrocytes, that these are very interesting cells, that they have immunomodulatory capabilities, but also they act somehow like neurons as well. They do transmit information from one astrocyte to another, just as neurons through synapses. But astrocytes, they use gap junctions to communicate through calcium waves, to communicate from one to each other. They're very territorial. They don't overlap in normal conditions, but in pathological conditions, they can have different morphologies, and their physiology could be altered. And then we have perivascular cells, which is a subset of microglia or potentially macrophages that are coming from the periphery and that stays in the perivascular system, and it would be close to the blood-brain barrier. So after potential injury or real injury at the periphery, specifically if the surgery is large or if it involves nerve damage, the first cells that will respond, changing the morphology, are microglial cells. So they will be larger, they will change the morphology, and they will change the pattern of molecules that they produce and secrete. Normally, what they do is they secrete pro-inflammatory factors or cytokines, and this is in parallel to pain-related behaviors. If the damage is larger, the blood-brain barrier will be broken physically. We will have some macrophages coming in the CNS. We will also may have T cells coming in, and this may happen also due to nerve injury, and this will interact with the other cells, with astrocytes, with microglia, with neurons, and this will also alter the milieu in the spinal cord. So this is just a representation of the very, very multipartite relationship of cells that we have in the spinal cord. We have the presynaptic neuron here, and the pointer is not working, but I will take another one and we'll see if it works. Works better. So we have a presynaptic neuron, we have a postsynaptic neuron, but these neurons are in intimate contact with microglial cells. We also have astrocytes that are also in intimate contact with the presynaptic and postsynaptic cells, and also with microglia. We have lymphocytes that actually communicate with astrocytes. We have perivascular endothelial cells. We have the astrocyte processes coming to the perivascular milieu, and we have perivascular microglia. So as you can see, any changes in these particular cells will provide information to the other cells and will interact, calling the multipartite communication, especially following nerve injury or other tissue damages. All right, so for our studies, we use a model, a very simple model of acute postoperative pain. This is the Brennan model. Tim Brennan just developed this model in 1997. It's simply an incision in the paw of the animal, a little irritation of the muscles, and then we close it with 2 sutures, very similar to minor surgeries in humans, but very, very, very minor. We stimulate the paw later on with mechanical filaments, with mechanical stimulation, with these filaments actually just pick the paw. And as I said before, if the inflammation is sufficient, a very small or a very weak mechanical stimulation will elicit the behavioral sign of pain reflexes. And that's what we observe. This is the baseline intensity that is needed to elicit pain-related behavior. After paw incision, 1 day after paw incision, this intensity is reduced, suggesting that these animals are displaying hypersensitivity, just as we do when we have a surgery. When the doctor, again, touches and examines the wound, when we woke up, when we mechanically stimulate the wound, it's painful, similarly to what we are observing in these animals. In the spinal cord at this time, in green, you have here IBA1, which is a marker for microglia, and in red you have GFAP, which is a marker for astrocytes. So at day 1, we observe already an increase in this marker expression. We have what we believe it is reactivity of microglia and astrocytes in parallel with the hypersensitivity developed at day 1. At day 3, the animals are still displaying pain-related behaviors, and we can observe how microglia is already very hypertrophic. The morphology has changed a lot. Astrocytes, they have lost their territorial distribution. They overlap to each other. They change the morphology as well, and this is also in parallel to the presence of the pain-related behaviors that we have after a simple incision. So all these changes in the spinal cord are happening by a simple incision in the paw. At day 9, these pain-related behaviors are resolved. They return to normal, and as we can observe, the expression of IBA1 or GFAP return to normal as well. So we observe a very parallel phenomenon in terms of pain-related behaviors and glial markers in the spinal cord, which has make us suggest something going on here. So when we administer at this point, at day 1, when we administer a CB2 agonist, a cannabinoid type 2 agonist in the spinal cord intrathecally, this is a CB2 agonist, we observe a reversal hypersensitivity in a dose-related manner. 2 micrograms or 10 micrograms of JWH was able to reverse the mechanical hypersensitivity after paw incision at day 1 after surgery. So suggesting again that the endocannabinoid system in the spinal cord is functional in paw incision model. In parallel, we measure the levels of glial markers. This is IBA1, this is microglia. This is a close-up of this image. This is the dorsal horn or the spinal cord, or the spinal cord. We observe that after the administration of vehicle and at day 1, we observe a very marked hypertrophy and overexpression of this marker in microglia. And then after the JWH, we observed previously that we reduce the behavior, so we reduce the hyperexpression of IBA1 or the hypertrophy of microglia. Similarly, we observe GFAP. This is astrocytes. We observe basal, very territorial demarcation of astrocytes in normal conditions. After paw incision, we observe that in some areas this territorial distribution is lost in astrocytes. It's very they're very hypertrophic after 1 day of surgery. And when we administer the drug that actually reduces pain-related behaviors, we observe a return to basal levels to astrocytes as well. So again, the normal resolution of paw incision hypersensitivity goes in parallel as well with the resolution of mechanical hypersensitivity when we manipulate the system pharmacologically, specifically with a CB2 agonist. Again, it strongly suggests that the CB2 receptors are functional in the spinal cord. All right, so the next experiments that we did was we would like to determine and characterize how the cannabinoid system is behaving in paw incision model. So first, we measure the CB1 receptor expression in the spinal cord at different time points and at different locations. In general, what we observe is that at day 1, day 3, and day 9, we observe a reduction of CB1 receptors. This is interesting because if we have less CB1 receptors in the spinal cord, we will have less circuit breaker signals, right? That means that the animals can be probably can have less inhibitory signals in the spinal cord. In other words, pro-nociception, or in other words, pro-analgesic behaviors or pro-pain-related behaviors. So CB1 receptors are reduced in the spinal cord. That's why we believe is that if we stimulate with CB1 agonists, with THC or other cannabinoids, we can reverse also the pain-related behaviors because we compensate this loss of signaling in the endocannabinoid system. CB2, on the other hand, we observe repeatedly that are overexpressed at day 1 after surgery at different levels in the spinal cord, and at day 3 as well, and this remains elevated at day 9. This is interesting because this could explain why the CB2 agonists, under normal conditions, they don't have any analgesic effect, but they become effective after point-incision. And this is in line with these findings. We have more receptors, they're therefore more sensitive to this. So a reduction of CB1, an increase of CB2. CB1 we found, as other people have found as well, that they're mostly expressing neurons. They're not expressing microglial cells. As you can see here, in green are the different types of cells, and in red are the CB1 receptors. So no microglial expression, mostly neuronal expression. No ED2 expression of CB1. They're just next to each other, but not co-staining. And then this is GFAP. We observed that astrocytes actually embrace and wrap some positive CB1 neurons, but they are not overlapping. So CB1 receptors are mostly or almost exclusively expressed in neurons. CB2 receptors, they are not expressed in neurons. We didn't find any CB2 receptor in neurons. As you can see, the abundance of CB2 receptors in the spinal cord is lower than CB1, much, much lower, but they're not in neurons. We found them mostly in microglial cells, as you can see at different time points. Similarly, we found them in ED2 cells, in perivascular, microglial, or macrophage cells. We did not find them in astrocytes, even though we can find some cells that are next to astrocytes, but they are not in astrocytes. So this goes in line with the first cartoon that I show, in which we have astrocytes together with microglia, together with neurons, etc. So we are finding these patterns as well. So CB2 receptors express mostly in glial cells, microglia, and CB1 receptor mostly are expressed in neurons. However, we have a reduction of CB1 receptor and an increase of CB2 receptors after point-incision. What happens with the endocannabinoid system, with the endocannabinoid levels, 2-AG and anandamide? So this is very interesting. After 1 day of surgery, anandamide, which is one of the major endocannabinoids, is reduced at day 1, and is reduced at day 3, and then returns to basal levels at day 9 and 15. Again, this goes in parallel with the induction of mechanical hypersensitivity. Again, if we have less CB1 receptors, if we have less anandamide, so therefore we have an enhancement of the nociceptive signaling. On the other hand, we have 2-AG, no changes at day 1, but then we have an increase at day 3 and at day 9. Interesting, because this could be explained, we speculate at this point, that this increase of endocannabinoids at day 3 and this return to anandamide at day 9 is probably driving the resolution of postoperative pain, because it goes in line with the resolution of postoperative pain. It's just an association. This is not functional, but just an association. This makes sense, because the endocannabinoid system has been involved, or it has been shown to be involved in stress-induced analgesia. Stress-induced analgesia, the classic example is the soldier that is in the battlefield. He gets shot, continue fighting, doesn't feel anything, suddenly he's weak, and then he finds himself that he's bleeding. He didn't even feel that he was shot because of the stress of the battle. This we knew that it was mostly due to endogenous opioids. And now we know, in 2010, it was described by Dr. Andrea Homan, that also endocannabinoids are involved in this analgesic response after doing stress. So it makes sense that the endocannabinoid system may be playing an anti-nociceptive role. So we speculate based on these findings that the endocannabinoid system is important for the normal resolution of postoperative pain. So to test that, what we did was very simple. we block the endocannabinoid receptors. We block CB1 and we block CB2 receptors. And what we did was very broad. We inject the CB1 and inject the CB2 antagonists, both together, and we inject it systemically. So what we did was we injected at day 1 after surgery, and we repeated the injection in the morning and in the afternoon for 9 consecutive days. 9 days because this is the time in which pain is resolved normally. So if we block the endocannabinoid system at this point, we expect to see if the resolution of acute pain is affected or not. What we found is, as you can see here, this is the 9 days of treatment. This is the animal that received vehicle, the group of animals that received vehicle. Hypersensitivity at day 1, day 2, day 5, day 7, and then day 8, day 9, returning to normal, just as we were expecting. The animals that received the antagonist, the blockade of the endocannabinoid system, they did not return to normal or basal levels after we stopped the treatment at day 9. So with these findings, we demonstrate that the endocannabinoid system is necessary for the resolution of acute postoperative pain. Interestingly, on the contralateral paw, this is the contralateral paw. This paw doesn't have any incision, any problems. It's a normal paw. The vehicle didn't do any changes in the basal behaviors, but the antagonist did produce hypersensitivity in normal tissue. That means that the endocannabinoid system in normal conditions is keeping the basal levels or the basal tone of nociception. It's important to understand that because that means that if we have a normal functioning of the endocannabinoid system, this is touch, is not painful, and this is painful. It seems that the endocannabinoid system is playing a very important role in the nociceptive system to differentiate what is painful and what is not under normal conditions. If we block it, this is altered, and therefore now we will find that innocuous stimulation is now painful. When we stop the treatment, this resolves, and then the endocannabinoid system works again. However, this hypersensitivity is already persisting after we stop the treatment. That means that at this time point, some mechanisms, whatever is happening, is already blocked. And the endocannabinoid system is important for stopping this process. And if this doesn't work, this acute pain will chronify, will perpetuate. And that is what we call transition from acute to chronic pain. So this is very interesting. So these are the 2 areas in which I wanted you to focus. We understand now that the endocannabinoid system is important for the resolution of acute pain, and also is important for the normal nociceptive tone. We measure glial markers. This is astrocytes. This is microglia. At day 9, we observe an increase in the expression of GFAP and an increase of IBA1. So microglia and astrocytes alterations are observed at day 9 following the blockade of the endocannabinoid system. Same pattern was found at day 15. Increase in GFAP and increase in IBA1. So the blockade of the endocannabinoid system is not only perpetuating pain-related behaviors, but also inducing perpetuate changes on, well, perpetuation is a really strong word, but I mean chronification of responses of astrocytes and responses of microglia. When we quantify that, we found significant changes only in astrocytes, but not in microglia, suggesting that astrocytes are more relevant in this chronification of pain due to the blockade of endocannabinoid system. So what we did was, okay, let's measure something else, because probably an increase of GFAP could mean nothing, right? It's just a hypertrophy. Astrocytes are just trying to compensate for something. So we measure a MAP kinase, P38. Phospho-P38 is important for the production of pro-inflammatory factors and is a very pronoceptive molecule. We observed that at day 15, the P38 was overexpressed in the spinal cord, as you can see in this close-up. P38 was enhanced after the blockade of the endocannabinoid system. And interestingly, this overexpression of P38 overlapped mostly with astrocytes and in some microglial cells as well. So P38 is enhanced after the blockade of the endocannabinoid system, mostly in glial cells. Okay, so what we did now is, all right, if this is true, if this is functionally linked, if we block P38, we should block the chronic hypersensitivity of these animals, but also we should block the GFAP overexpression. And that's what we did. We tested with propentofilin. Propentofilin is a glial modulator. It's a glial modulator that is known that reduces GFAP and is known that reduces P38 phosphorylation, both. So it was a very, very nice tool for us to use. So we administer the drug, the antagonist at day 15. We take the animal and then we measure, then we administer in some saline, which is the vehicle, or propentophilin, and we observe in the vehicle group an increase in GFAP, and then in propentophilin we blocked the GFAP overexpression. Similarly, we block the overexpression of phosphorylated P38 in the spinal cord. When we measure then the behavior, this is what we observe. This is day 14, day 14, day 15, day 15, after the blockade of the endocannabinoid system, basal levels of pain-related behaviors, hypersensitivity, chronic hypersensitivity. We inject propentophilin here, we inject propentophilin here, and we observe a reversal, at least partial, of the pain-related behaviors in parallel with the inhibition of P38 and the inhibition of astrocytes. So we conclude then that the endocannabinoid system signaling is necessary for the resolution of postoperative pain and for the basal tonic nociception. And also we conclude that the endocannabinoid signaling may perform these functions by limiting spinal astrocyte expression of GFAP and also for the phosphorylation of PP38, mainly on astrocytes, but also in microglial cells. So we further hypothesize, we speculate that probably the patients that develop chronic pain after major surgeries, remember, 50% of patients, for example, with mastectomies will develop chronic pain. This is interesting, you think, because why 1 out of 2 women will develop chronic pain after mastectomy? What happens? What is the difference? Why one does develop it and the other don't? We don't know. We speculate that probably the person that actually developed chronic pain after mastectomy is due to a malfunction of the endocannabinoid system. So interestingly, this is a very testable hypothesis, right? So our data make us to speculate that. But what does this mean in reality? What does this mean in reality in the real world, in clinical, in patients? So we tested propentophilin, that is a very good known glial modulator in a clinical trial. And as I said before, it failed. Others tested also endocannabinoid enzyme inhibitors in humans. This inhibitor was proven to enhance anandamide in humans, but it failed to reduce osteoarthritis pain. So my endocannabinoid system theory is breaking apart because the clinical trials, again, are failing. I told this already to my students, but they keep coming to my lab. And these are the 3 clinical trials of the 3. These are the only 3 clinical trials, at least up to 4-6 months ago. 3 major glial modulators that it has been studying for 1 decade or 2 decades with amazing data in animal models, all of them. In my laboratory, we tested propentophilin. My former mentor was the discoverer of propentophilin. And it works beautiful, reducing glial changes and also reducing pain-related behaviors in rodents. So these are the randomized, double-blinded, placebo-controlled, you name it. I mean, these are the different drugs, paradigms, 8 days treatment. Propentophilin was given actually 6 months. This guy, the CCL2 inhibitor, was given for 28 days. And this is what happened. Pain treatment efficacy, none, none, none. For some, minocycline is a microglia inhibitor. They didn't use any drug levels on function assessment. In the propentophilin, we measured plasma levels and drug was detected. With this other drug, plasma levels of drug were detected and also a reduction of monocytes counts were found in the drug group and not in placebo. So it seemed that these drugs were actually in the system and they were supposedly to work at these levels. But as we said in the discussion, probably some interpretation of the data or the clinical design was not correct. But anyways, 3 clinical trials, we should be really bad if we actually design really badly clinical trials, right? So anyway, as I said before, the CB2 agonists, the cannabinoid type 2 agonists, they were also stopped for clinical trials because they didn't show any efficacy, even though I showed very beautiful that they resolve pain-related behaviors in animals. The endocannabinoid enhancers, the FA inhibitors, they fail in pain osteoarthritis in patients as well. So 5-6 clinical trials in glial modulators that they're not working. So, or either we are really bad scientists that we don't know how to design clinical trials, or there is something wrong here that we don't understand. So Thank you to my student. This is a very bright student. He was an undergrad when he was helping with this studies. He did a PhD in Oxford. He was at Dartmouth. He did also a PhD at NIH and Oxford combination. He's in Harvard Medical School now. Amazing guy. And then also thank you to all my collaborators and my funding sources. And I had to catch a plane in 1 hour and 50 minutes, but I have time probably for a couple of questions.
It's a very interesting study. My question is, for the clinical trial, the chronic pain patients, what kind of